All right, we're doing good. So we'll just take a few minutes here and let people filter in. I don't expect to have a crowd here because, okay, some esoteric topics, kind of narrow focus. It'll still be fun, right? At least I tell myself that. Oh, just as I start this, I get an alert that Mikey has come out with a new video. Oh, well. Yeah, it happens. It's okay. Cryptic is expecting fun. Yeah, Mikey. Everyone knows Mikey. Let me see what's it called this time. He does Film Joy. Yeah, he does fun stuff. Very enthusiastic about movies and so forth. Somebody read the choanoflagellate paper in lab meeting. Wow, okay. So you're going to be able to argue with me. Maybe, I don't know. Or you'll agree with me, or I'm just going to be reciting the received wisdom. Okay, well, I will start babbling. So my goal here is just to work myself back up into this YouTube stuff because I have been distracted. I have had all this work I've got to do, and it's just piled up on me. And so I've been bad about YouTube. I got a couple ideas for things I can do on YouTube, but I haven't been doing them because spiders. Yeah, somebody asked about spiders. No, I'm not going to talk about spiders today because spiders scare people away even more than technical subjects in biology. It's just bizarre, but here we go. Okay, so what do I want to talk about? I put up this list of papers, and I thought I would just start talking about them. The list of papers simply to give some structure to what I'm saying. So please do interrupt with questions or comments. Tell me stuff you're into, and that's all fine. I'm also imposing a strict limit of 1 hour on my conversation. I cannot keep students who are paying for my class to be entertained for a whole hour, and to say, well, let's just go on for a couple is, no, I'm not that arrogant. Okay. So what am I going to talk about? Oh man, I got all these screens of things here. Let me just shuffle stuff around a little bit. Okay. So I gave you guys a list, and I said I'd talk about a couple of interesting papers that are out. And the first one I chose was this one by Christy Wilcox. So Christy Wilcox, I know Christy Wilcox. She's a smart person. And one of the things I really liked about this particular paper titled "Can New Species Evolve from Cancers? Maybe. Here's How," is unlike many of the pop science articles I run into, she actually applies some skepticism, some critical thinking to the paper. There are so many pop science places that we turn this into, oh, organisms arise from cancers, and oh, isn't that exciting? And this paper is actually pretty honest in dealing with it, that this is a controversial proposal. It's not accepted. There are some real problems with this particular hypothesis, and we'll get into those. So, what is the paper about? Well, there are these interesting organisms called myxosporins. I first ran into myxosporins because I was working on zebrafish And the problem with there is that myxosporins cause a disease in fish. They're parasitic. They can infect fish. Various things to the cartilage. They infest cartilage. That's where they prefer to go. But they can also cause scar tissue in hearts. They also populate the brain, and they cause a disease called whirling disease because the poor fish just does this as it tries to swim along, and it's more susceptible to predation. And yeah, it's a bad, terrible disease. And the cool thing about it, though, is that it's caused by an infestation of these myxosporins, which are multicellular. What we call them, they're multicellular, but they're very, very parasitic, and they got a reduced genome and all this other cool stuff. They are thought to be cnidarians of all things, so they seem to be. Oh, somebody's getting lag and stuttering audio. Maybe it's just me. Maybe I'm stuttering. Anyway, myxosporins are they're multicellular organisms that produce these diseases by infesting fish. They also have a complex life cycle, which is kind of interesting. So they infest annelids first, and then secondarily infest the fishes. So they're not real simple organisms. They're real metazoans, which have adopted a parasitic life stage and have complex life cycles. Okay, so the paper, what is it about? Well, there is a hypothesis. For a long time, it was a hypothesis simply that they were derived from cnidarians. So that was kind of a shock when it was first proposed. Now what this guy is saying, what these people are saying, is that they are descended from cnidarian cancers, which is kind of weird. All right. So the idea is that long, long ago, cnidarian had a cancerous growth that led to excessive proliferation, like we see in cancers. And these cancerous subthreads coming off of the cnidarian adopted a secondary lifestyle. They're now independent. Well, no, not independent. They're independent of the cnidarians. Now they require the parasitizing things like fish and annelids in order to survive. Okay, so what's the evidence for this? And that's where it gets wobbly. Okay, the hypothesis, the very speculative hypothesis that arose. And the original authors of this hypothesis proposed that what you would expect to see if this were true, if it actually arose from cancer cells, is that you'd see some of the similar losses of genes that regulate growth in the cancers. So what they do is they then go and look at the genome of cnidarians and try and see what's missing and see if it compares to what you find damaged or missing in cancer cells. And what do they find? Well, they find that there are some interesting losses in the myxosporin genome. Then in particular, they saw things like losses of p53 and associated proteins, which you also see in cancer, that p53 gets knocked out. They saw that some of the death proteins, the genes that are responsible for apoptosis, are missing from these cells, suggesting that, well, again, like cancer loses those genes. It's a way to minimize the ability of the host to trick them into committing suicide. So that's really suggestive. But as Christy Wilcox points out, and as the reviewers of the paper also point out, this is a provocative hypothesis. but it's really speculation at this point. And that they have adopted a parasitic lifestyle is going to be accompanied by a loss of genes. That's really kind of standard. So to say that these cells have been found to have lost some genes is not definitive. It doesn't simply say that, well, then they must have come from cancers, because parasitic organisms lose lots of genes, just that they've lost some of those. What's interesting about that is that, in dealing with creationists, for instance, they'll often tell you that scientists are out there suppressing these ideas. The reviewers for this paper all say, I don't believe this. I don't think this is true. I don't think the evidence is good enough to support it yet. But it's an interesting hypothesis, so publish it, and we'll sort it out later. We'll let people try and do analyses to figure out. So that's a really good suggestion. And it sort of tells you how science actually works, is that we love provocative hypotheses, but we have expectations for evidence to back them up. And what's interesting about this proposal is that I don't think their work is adequate to demonstrate the validity of their hypothesis. It's a big hole right there, a big gap. But this is the kind of thing where you could expect somebody to do a deeper analysis of the genome of cnidarians and come up with maybe better evidence or counter evidence. I'll just say that one of the reasons I don't quite believe it is, as I said, myxosporins have a complex life cycle, and they're fairly sophisticated organisms. And no, I'm sorry, but myxosporins aren't as screwed up as cancer. Cancer cells are really messed up cells. And these myxosporins are viable and have good lifestyles, and even if it's parasitic lifestyle. So I don't think that's very likely. Okay. Do people have questions about that? Anyone have anything interesting to say about it? Oh, I'll set it aside. Okay. But look it up anyway. It's a good example of how to do popular science right. How to explain how things actually work. I give Christy Wilcox a thumb up there. Let's see. So let me just look at the comments over here. Everyone, let's see. Okay, a couple questions. If this hypothesis were to pan out as true, would these new organisms be cladistically classified as cnidarians? And yeah, they are. They belong to the cnidarian clade, but yeah. Let's see. I got a request to join. Oh, Nestle would be a good one. Let me see. This new YouTube setup is so confusing. How do I invite somebody to a stream? I don't see anything about that. No. So my technical incompetence is going to get in the way of inviting other people to join in. But please do make comments and let me hear what you want to hear about. A question about cladistics. What is my question? I thought I just answered that one. So these myxosporins would be members of the cnidarian clade. Is that the question? Sure, they would. Okay. Lots of people want to hear about Ediacaran embryos. I bet you that's because Carl Zimmer wrote about them in the New York Times, isn't it? Yeah. All right. I will get to that. Let me take a moment here, though. So, we got more questions about joining. Let me see. This is, So this is using the new Google Studio, YouTube Studio, and it's a horror. I don't know why they keep changing things, but they do. I don't see anything where I can let people join in. Okay, yeah, you don't see anything from the New York. That's a real shame because I don't like the New York Times either. And Carl Zimmer publishes in there, and he always does really good stuff. So let me talk before we get to the Ediacaran embryos. I'm sorry, I'm going to make you wait. I want to talk about this other paper because it's also relative to a lot of my interests. And this is the one about how many genes do cells need, maybe almost all of them. And this is something I try to drill into my genetics students, that we have this association of genes with phenotype that's constantly reinforced by simplistic introductions to genetics, that we're constantly talking about a gene for X when there's no such thing. It's always much more complicated than that. So this is a paper that talks about some yeast studies, but I've seen similar studies in Drosophila. And here's the basic idea, that long ago, the way we started studying genetics in Drosophila and other organisms was you look for mutants, try and knock out individual genes, and you ask, what is the effect of that mutation on the organism? And this is what leads to the idea that that particular phenotypes are the product of individual genes. This isn't a valid conclusion, however, because what we've found over the years is that there are an awful lot of genes that you can knock out and you see no phenotype. All right, so that's sort of suggesting that there's a flaw there, unless there's an awful lot of genes that just simply don't do anything. They're lazy, just lying around in the genome, not accomplishing anything. So there were a number of studies that were done where they do something a little bit more different. So you've got this gene that you've knocked out, and it doesn't seem to do much of anything. And you get this other gene that you've knocked out. It doesn't seem to do much of anything. What happens if you combine those two mutants in a single organism? And what you find is that sometimes the product of that cross just goes bleh and dies and does horrible things as really blatant phenotypes. So many of the phenotypes are the product of two otherwise fairly subtle genes that are interacting to produce the phenotype. And you knock out just one and the other one fills in for it. Or it could just be that the phenotype is so subtle that you don't recognize it in the single mutant. You got to have multiple mutants to make the phenotype strong enough that you get the problem. So what this is about is that these studies that were done in yeast. Oh, there we go. Somebody in the comments says, yeah, somebody's... So he said, I'll read this because it's relevant. I work in a yeast lab. There was a recent coup that apparently the genome deletion collection is full of compensatory mutations. So the gene deletions don't actually do what you think. Yeah. So that's a flaw in this particular work, but it's still the case that what we find is that if you're working with an organism that has 6,000 genes like yeast, or 20,000 genes like humans, some of them may actually be inessential in the wild type context or in a normal situation. But if you mutate something else, you may find that then you need that previously unnecessary gene. Or it may be that there's some synergistic interaction between multiple sets of genes that are necessary for the phenotype. So this is a study where they did this with yeast, just basically combining mutations 2 by 2 and discovering that there were new phenotypes emerging from these. and then also gathering together three genes, three mutations at once, and seeing what happens. And again, you see it going kerblooey, that the organism takes these, what we thought were inconsequential, fairly neutral or fair little genes, and in combination cause all these problems. This has led to this idea that genes are omnigenic. That is, that every gene is essential for the function of the organism in some way, that they work together to generate the phenotype. So that fits with my experience. That fits with what I learned from working with Drosophila. That you need you need redundancy, you need some duplications to reinforce particular phenotypes. And what this paper is arguing is that what that may mean is that there's no such thing, or they're very rare to find inessential genes in an organism. They all contribute in some way. So you can't just, you can go and start deleting genes, and what you'll find is maybe you get negligible effects at first, and then you hit a threshold where you've knocked out a lot of the functions of the organism, and you get death or obvious distinct phenotypes out of it. Yes, so we've got another comment here. When Craig Venter tried to pare down a bacterial genome, the one-by-one deletions were completely insufficient to tell the minimal set. Right. So you get this idea, well, I got an organism with 600 genes. Can I just start knocking out one at a time and find out what the bare minimum you need for a functioning organism might be? And you suddenly find that, well, you knock out this one, it's fine. But when you knock out this one and this other one, it's no longer fine. So it's not so simple as we once thought. Cryptike is saying, I was wrong. It was 2 by 2. I misremember. No, the original experiments were 2 by 2. Okay, they were knocking out pairs of genes. And then what this new study was doing was knocking out trios of genes and seeing an even greater effect. So, yeah, this is kind of, I'm going to be teaching genetics in the spring. So this is really relevant to what I'm going to be talking about there, because I run into this every year. I start talking to my students by teaching them, simple Mendelian genetics, and I got to be constantly telling them it's going to be more complicated than that. And eventually, we're going to get into all these static interactions. Yeah, I can make students cry every year just by bringing up epistasis. I'm sorry to say, it's so much more complicated than most people think. This is also a reason why I really resent some of the racialist arguments out there that are trying to argue, yeah, there's something wrong with the Black genome or something especially good about the Asian genome. No, you can't do that. Humans have been interbreeding for thousands of years, and we've all got these genes floating around. We got the same genes. We got similar alleles floating around. So it's just not a good thing to get trapped in this ancient, simplistic Mendelian system of thinking. And okay, let's see. What am I going to talk about next? Oh, let's see. Nestle is still trying. Can you copy the link that you can see on top of the screen of the broadcast and send it to me? Oh, I got different. Oh man, this is a mess. I'm going to figure this out later. Let's keep going, because I'm only going to talk for a half hour more. I want to be able to get some questions from you. Oh yeah, and Diane Bruce mentions the infamous gauging. Yeah, that's another great example Why would you think that something as complex as sexual preference, with all of the layers of genetic and cognitive activity that are going there, would be the consequence of a single trait? It's just silly. But and I really think that part of the reason people aspire to these sorts of things is because of the way we teach students about genetics. They get this simplistic version in high school, and that's all they know. Yeah, they think everything can be solved with a Punnett square. No, it can't. Yeah. Okay. So, what was I going to talk about next? What's next on my list? Oh, shall we talk about choanoflagellates? I've been fascinated with choanoflagellates for decades now. They're extremely cool animals. And oh man, I'm just noticing I was going to show some videos, and not only do I not have a way to invite anybody to this stream, but there's no way to show a video in another file. How annoying. Okay. Well, I will just have to do it with hand gestures then. We'll see. Okay. Okay, choanoflagellates. What's so cool about choanoflagellates? They are initially described as single-cell protists or protozoa that that they're known to be quite capable of living independently as single cells. And so, again, that's how they were classically described. They have a roundish, ovalish cell body, and then the distinctive thing about them is they have a collar on one end with flagella coming off of it. So choanoflagellate, choana means collar, so they're basically collared animals with these whip-like flagella extending out in one direction. They are filter feeders, so they basically swim around, the little flagella waving around here and collecting bacteria that are then filtered back towards the cell body. They were thought to be a really interesting and potential intermediate in the evolution of animals because they fall into a clade related to animals. Among other things, they contain receptor tyrosine kinases, these cell surface molecules that were once thought to be unique to multicellular animals. You don't find them in plants, for instance. And choanoflagellates have these RTKs. Also, that when you look at the cells, look at their morphology, they are strongly similar to the cells that you find in sponges. So sponges also have these collar cells in a matrix that they use to generate currents that flow through the tubules inside of the sponge. That's basically all a sponge is a bunch of collar cells linked together by some other cells with mesoglia glue underneath them, making a kind of efficient blob for sucking in water and running it past lots and lots of flagellae so that you can extract all the nutrients from the water. So that's what a sponge is, and sponges are animals. So the idea was that they're related somehow. The recent thing, though, I shouldn't say recent anymore because I'm old and everything I thought was new is now old, but relatively recent is the discovery that they have these interesting cooperative behaviors. So sometimes you go out looking for choanoflagellates and you find they are not all solitary cells swirling about in the ocean or freshwater. They're all over the place. What they sometimes do is they link up and they form groups. They can form a kind of raft of choanoflagellates all linked together. And what they can do then is more efficiently filter bacteria out of the water. They can also move in the water column. So one of the things they found. And sometimes what they'll do is they'll separate. They'll do that in feeding. Sometimes they'll do cooperative feeding, and then they will clump together and orient themselves so that they can move in the water column more efficiently. They have better locomotion when all the flagellae are all aimed in the same direction and all going the same way. So yeah, question, they get sticky. Yes, they get sticky. They adhere to one another and form the structure. Okay, so that's cool right there. So it says you got these organisms that look very similar to those you find in sponges, and then you find that they are not solitary in an obligate way. They can cooperate and form simple tissues that can do interesting things like more efficient feeding or more efficient locomotion. What's discovered in this research article is they found, they went down to Curacao. Why can't I be working on research that let's go down to the Caribbean, the tropical Caribbean, and collect animals? That's, oh man, I missed an opportunity there in my career. Anyway, what they found is they went around tide pools on this island, and what they found were these really interesting choanoflagellates that formed these cup-shaped structures. So let me see if I can show this here. Yeah, there's the paper. Wish I could show you the movie. I'll figure that out eventually. Anyway, so what do you see here? Well, there's what you see in the microscope, and there's a nice movie of these. And what they do is they form these kinds of cup-shaped structures where all the flagellae are pointed inwards. And that's a good feeding strategy. So they're much more efficient at filtering out food particles from the water in that configuration right there. But then what they can do is this. They can invert themselves. And all the flagellae are now pointing outwards. You see all those right there. And when they do that, they're better at swimming, but much less efficient at feeding. So here you got this animal that responds to its environment and can be either in a feeding specialist mode or a locomotor specialist mode for getting away from things, for instance. That's pretty cool right there. But then they figured out what the trigger is, and it's light. They are light sensitive. So when you change the light level, they respond to that by inverting to switching into one of the other modes. So these are really simple cells, and here they've got a light-mediated reflex, and they've got multiple modes of organization for multiple behaviors, which is just kind of awesome, right? Yeah. So, oh yeah, and they also, caffeine has the same effect. They're actually using a cyclic GMP-mediated photoreceptor system with retinal and an opsin, just like us. You know, our retinas, they have this little molecule called retinal that changes conformation when exposed to light, that is bound up to a protein called opsin. That's the case here, and then further, their opsin is coupled to a signal transduction mechanism using phosphodiesterase. So, yeah, it's all very familiar. So this is pretty cool. This is a sensory modality. They're able to sense their environment. They're able to respond to that environment. They use cooperativity to form multicellular sheets. So we could ask, why did multicellularity evolve in the first place? And choanoflagellates give a pretty good example of an organism that could be living as solitary individual cells but finds that they are selected for favorably by forming these cooperative masses, which is kind of awesome, especially for socialists who want to see more cooperativity and less competition in our world. That this is an example where it says, yeah, that sometimes works pretty well. I am not going to fall for the naturalistic fallacy, though, and say we should cooperate because choanoflagellates do. Okay. Any people have questions about that paper? No? It was so obvious, isn't it? Humans, cooperative masses. Yeah, we're cooperative masses. Look at us. We're trillions of cells, and they're all cooperating to function. Okay, another question: do they subset by function as slime molds do? Oh yeah, that's an important distinction between colonial organisms and true multicellular organisms. Is true multicellular organisms get into specialization, so they take cells, they specialize them to do particular functions. So humans, for instance, we have some of our cells are making brains and some are making muscles. Those are all dead-end jobs because they're not going to get passed on to the next generation. And some lucky cells get to be gamete producers and ovaries and testes. And those are cells that are going to go on and reproduce themselves. So your brain has dedicated its existence to making sure that your genitals produce gametes. It's one way to look at it. They've sacrificed so much for my testes. It's kind of charming. Okay, the question here. I can't help but wonder if some of these microbes, choanos, Foreshadows, Ichthyospora, used to be more complex, but animals got so good at multicellularity, they excluded middlingly complex multicells. That is a good point. Yeah, that's actually going to be relevant when we talk about the Ediacaran paper. There's an assumption here that choanoflagellates, because they look like simpler versions of multicellular cells, are precursors, and that is not necessarily true. That they could be descended from more complex organisms that have disassociated to fill a different niche in the environment. Just like the myxosporins, right? So the myxosporins, remember, were cnidarians that have now adopted this parasitic lifestyle. Who knows? Maybe choanoflagellates are descended from some more complex organisms that found more success in living independently. Maybe choanoflagellates are just disassociated sponges. I don't know. So there's some ideas there. Let's see, another question. I've heard before that ctenophores are basal rather than sponges. Yeah, that I've heard the same thing, that ctenophores are more likely to be similar to our last common ancestor than sponges are. The sponges, in a lot of ways, are weird organisms. That doesn't exclude them from being at the base of the animal tree, but it sort of suggests we'd rather see something a little more basal there. Okay. I was under the impression that sponge specialization was temporary, though, that if the sponge is physically injured, that the exposed non-flagellar cells would grow flagella. Yes, there's, let's see, how do you get into this? You're sucking me into a black hole already. Yeah. That this is a thing about a lot of multicellular organisms, that some of us retain the capacity to regress to a simpler state. Okay, so think like in a salamander, if you chop off a limb, you get a blastema. A blastema is basically a collection of cells that are reverted to a stem cell-like state to regenerate the limb. So yeah, that sponges can do that is not a strike against their origin. It's just an interesting fact about sponges. Okay, Professor Frank says, hello from Montreal. Hello from Minnesota. All right. The next, let's talk about, this paper. A lot of people have heard about it because Carl Zimmer talked about it, and he did a good job talking about it. This is an interesting paper. I have some quibbles with how they're interpreting the data, though. What's interesting about it is these are Ediacaran fossils, and they have this very suggestive appearance in that they form these convoluted masses of cells, fossilized cells, and I can believe that those are cells, and I can believe that these are individual organisms. Among the things that leads me to believe that is you look at these cells, and they are very similar in size, for instance. And this is about what you'd expect, that if you're looking at something that's non-artifactual, you're looking at an animal, you would expect to find that the cells have similar proportions. You wouldn't find any grossly small ones, grossly large ones. So these really look like some kind of multicellular organism of some sort. So a lot of this paper is dedicated to analyzing these cool looking fossils. And let me just skip ahead to what's a good figure here to show you. Yeah, like this one. So there's a couple of features that are kind of neat. Like I said, the cells are uniform in size, relatively uniform. And it's also surrounded by a thin sheet-like capsule. So these are basically balls of cells that are surrounded by something like a chorion in a modern embryo. So right there you can see this is the pattern that they see, where those gray areas are what you would call the cellular regions, and then there's these spaces. The thing about this that bugs me is, well, there's a couple things that bug me. Let me first point out the really big one that bugs me, and that is this is done after the fact. This is presented as if it is a time series in the development of this organism, and they can't do that. That's a hypothesis, okay? That maybe it's a good hypothesis, but the thing is, they are going out and collecting at this, and at the site that's full of phosphatized embryos, and they're pulling them out. They don't know what the developmental stage of each one is, but then they are assembling it into a sequence. That's where it really bugs me, okay? Because yeah, sure, I can take a random arrangement of cells and I could shuffle them around and say, okay, this is the order at which they develop, but is it really? I'm fitting it to a preconception right there. Further confounding all this is yeah, this thing up here. When they do this, what they also find is that over their speculative time series, and I emphasize this, it's a hypothesis that this is a time series that they've got them arranged in, okay? Over the speculative time series, they see something really weird, which is the embryos get larger as their proposed developmental series goes on. That just is not how embryos work. So embryos are set up with a set of maternal investments. They've got, more modern than Ediacaran organisms would have, like, for instance, yolk or some other nutrient supply. And evolution or development then proceeds by subdividing those into cells. So you expect the cells to get smaller and smaller and smaller as time goes by, while the net mass is going to stay the same. And they see this in the time series. Yeah, the cells get smaller and smaller and smaller. But at the same time, what they're also seeing is that the whole embryo is getting bigger. How? That's my question. That shouldn't happen unless they are somehow absorbing nutrients from the environment. But it's an embryo. Yeah, if this were a larva, this is where it would fit in. Is if we had a larva feeding stage, then maybe that's what's going on. But you look at the structure that they've got there, and how does that fit with a functional feeding system in this organism? I just don't see it. Of course, biology is smarter than we are, so maybe there's something going on here that we just don't know about. Okay, another thing about this that bugged me is, all right, so they've got a time series, and I can show you this here. This is kind of their time series of this. And what they argue is that there are cavities inside the embryo that are slowly getting filled in by the developing cells. So, like look at there. That could be like, a blastula stage embryo, if, to compare it unfairly to modern embryos. And what they see is that that cavity gets smaller and smaller as our hypothetical staging series goes on. But then there are other strange things. I mean, look at this weird kind of convoluted surface. We don't see that in modern embryos either. That as embryos develop, they tend to form, a minimal energy surface. Go ahead, talk to Darcy Wentworth Thompson, and he'll explain that to you. But these are this weird raised convoluted pattern, and then you get some that look like, or you look inside and they look like this. I don't know how this all fits into an embryonic series. I think they assembled their series by looking at the relative proportions of the cellular material to the cavities, the spaces inside the embryo, and hypothesized it fills up as time goes by until you get a final embryo. But again, we don't know what the final result is. It's just kind of hanging there. And the fact that the whole embryo gets larger and larger as this proceeds, I don't know. That outer shell, that chorion-like outer shell, is also a problem here because it's encapsulated, right? That sort of says you're not going to be able to get much way of nutrients unless it's just absorbing from the environment. That would be weird too. So yeah, I'm looking at these three-dimensional reconstructions, and it does not fit with any modern organisms that I know of. That's not a problem because, well, all right, it's an Ediacaran organism, so who knows what its function was or what the process of development was. But then how can you use your understanding of modern embryos and how modern embryos progress to build a series out of a collection of different sizes of cells? Yeah. Okay, we get a comment here. It's thought that macroscopic Ediacaran fauna just absorb organic material from their environment. And that's a reasonable hypothesis. hypothesis. Sure. And, when you look at some of the larger Ediacaran organisms that are sheet-like, are lying on the surface on the bottom floor of the coastal regions, yeah, you can see they're maximizing their surface area to extract nutrients from their environment. I just don't know what the niche for this thing would be. Why do you have all these bulgy bits that increase the surface area of the organism? Maybe that's a hint. Maybe they are absorbing nutrients through that greater surface area. And that's the whole purpose of this convoluted pattern of development, is just to make a ball of cells with complicated shapes that then directly absorb things from the environment. Again, the capsule gets in the way of that. Why are they surrounded by a sheet-like capsule then? I don't know. You might also expect, okay, you got this. It'd be really nice to see if there are cilia of some sort surrounding that. That would also be a factor in improving the feeding. If we accept their idea that this is a developmental series, which I don't entirely accept, okay, I have questions about that. That implies that this organism has to be feeding continuously because it gets larger over their developmental series. And okay, it's just a whole set of problems there. All right, do we know what kinds of proteins might make up that capsule, and some idea how permeable it might be? No, we don't. It's phosphatized. It's all been completely replaced by this stuff. So we don't know the exact structure. I would like to have seen more ultra-structural analysis as well as they can of, okay, so here's some examples. So there's our blobby little organism, and you can see the capsule surrounding it, right? It's just cracked open right here. I would really like to know what the edges of that capsule look like. It may be that we've reached the limits of resolution of the fossilization process, so it won't tell us anything. But still, I don't even, this could be a cellular capsule for that matter. For all I know, yeah. Because like mammalian embryos, we form this blastocyst, and it's got this external layer that's going to contribute to the placenta and the extracellular membrane and all these sorts of things. And in us, it's cellular. On the other hand, you look at something like a fish, and it builds a chorion that is protein-based. It's just a really proteinaceous shell around it. And of course, other organisms can build really elaborate external shells. Look at chicken eggs. Chicken eggs are not, the egg itself is not cellular. It's deposit calcium, although underneath it, it does have a thin membranous layer of cells. So, yeah, what can I say? That I know everybody wanted me to talk about the paper, and I'm less than enthusiastic about it. It just raises too many questions for me. Because yeah, it's just so suggestive, but I don't know whether they got that. For all I know, these different organisms that they have assembled in the developmental series could be, 8 or 10 different species that they've assembled in. You cannot determine that. So, yeah, how to interpret that? Okay, physicists get to work, make a time machine. Once upon a time, I thought it'd be really cool just to go back to the Cambrian. Now, apparently, I have to go all the way back to the Ediacaran in order to solve this puzzle. So I'd really like to know. Okay, do we have questions that have accumulated here? Does the arrangement of cells look like the arrangement of cells in an animal embryo, even if the large structure isn't the same? Oh, that's a good question. When I look at these, no. Okay, so like that one right there, look at that three-dimensional shape. Uh, as I mentioned earlier, when you look at embryos of extant organisms, they tend to reduce energy and conform to the shape of the tissue. I'm thinking of a frog egg. A frog egg would be relatively smooth on the surface here and would maybe have a point of involution for gastrulation, but that's about it. And they have a cavity on the interior. It would be a discrete cavity that you can recognize. So, no, I'm looking at that and I'm saying, no, that does not look like any embryo I have ever seen. It's really weird looking, which makes it interesting, but I just question their assembly of these weird structures into a developmental series. Okay, got a question here. Could you briefly tell me what's going on? All I know from the title is it's about science. Oh, actually, it's almost 3 o'clock, and I'm going to stop pretty soon here. Yeah, what I think is, this is just me having read some papers this week and getting on YouTube and babbling about them while slurping down Coca-Cola. That's the whole thing. Let's see, another question. So do you think these are ultimately more likely to be adults or something entirely other than embryos? Oh, tough questions. Oh, okay. What do I think? All right, well, it depends on how we interpret the data. All right, so if we accept the conclusion or the hypothesis that they have a developmental series that goes from small to large, that would tell me that it's not an independent organism. It's not an adult because of that increase in size. Yet, if I reject that, then I have to question, why did you put those in a developmental series? You're making the assumption that these are embryos from the space-filling characteristics of the cells inside that shell. All right, but I don't know. I just don't know. Yeah. Okay, are these from creationists? No. This is serious work. This is interesting stuff. It's an attempt to categorize and describe a set of very curious fossils from a Chinese formation that phosphatizes the fine structure of the fossils so that you can actually see details of the structure. So that's not creationist at all. That's serious science. So, yeah, could Volvox be relevant? Could we be looking at colonial assemblies? That's a reasonable possibility. So, because we're seeing, oh, this is tricky. Because we are seeing in each of these organisms, similar sized cells throughout that don't show any signs of specialization. That's another good reason to suggest these might be embryos. Okay, so we're looking at blastula stage embryos maybe, or gastrula stage even. Although at the gastrula stage we'd start to see differentiation into different cell types. But anyway, so we're looking at an early embryo that's dividing multiple times. That's one hypothesis, and I think that's what they accept because. Yeah, but putting them together in a developmental series that doesn't go anywhere, that doesn't come to any good conclusions, that's tough. Yeah. But then I guess it fits with what we know about the Ediacaran animals, that they're really cryptic and we don't know quite what they are. And here we're finding what might be embryos, might be some really peculiar adults with a strange lifestyle. And we don't know what it means. Yeah, that's science for you. Okay, I'm not going to get to the last paper because it's 3 o'clock now. I really I want to restrict these so they don't go off into the deep end and get all crazy and annoying at the end, and also so people can watch them if they have limited time. Do you have any other questions, anyone? Speak now or forever hold your peace. I'll try to do these more regularly in the future. As you can tell, this is just kind of stream of consciousness. Hey, here's some stuff I read. Let's talk about it. And yes, I will work on figuring out how to bring other people in to contribute to the discussion, like Nestle wants me to do. I don't know. This live studio is just weird, and I blame Google. Okay. Are you? It's so Nestle is asking if you're going to invite others. Or I'm stopping after the last paper. I'm stopping after this paper. I'm not inviting others because there is no obvious mechanism for doing that. Let's see. Here's something. There, I see a participants line, but no, it doesn't tell me how to invite anyone. Okay. Sorry. I will try to figure this out. I got to learn more about Google's new live studio, and we'll puzzle this out. Okay, it's been fun. I like just talking to you guys about what's in my head and hearing from you, and I will try to get more engagement in the future. So it would also be fun if you're doing it more often to be able to select a few related to that's what this is about or whatever. And yeah, you mean you want coherence? Oh, that I don't read coherently. I just kind of browse around. I find stuff that's interesting. The only attempt at coherence I made here is that I avoided talking about spiders. I feel bad about that, but maybe sometime I'll have to talk more about spiders, but not this time. Trying to persuade people this is not a terrifying thing to do. Okay, so let me end it there. Oh, somebody says, I think you just have to give them the link and they click and they're on, but I don't know. Huh. Okay, let's say I'm going to end the discussion at this point. We're through, but I'm going to just do an experiment, and I will email Nestle this link I just copied. Let's see. Oh, I bet you don't need all of that. Okay. Presto, I just messaged Nestle 20 with a bit of the URL. And we're just going to, this is just, again, an experiment. Let's see if we can get this to work. Yes, Nestle, check. See if that link works for you and let me know. What is the control? What are you, a scientist or something? Yeah. Okay, Dan Bruce likes photographing spiders. So do I. My plan for the afternoon is my wife and I are going to get together and do a little spider safari around the house. So we're going to just see what we can find. She's been finding a couple of nice pterodactyls around the house, and she says there are swarms of ulcids all over the house. So I got to go looking for those and get some pictures. Okay, let's see. All right, I sent something off to Slig. Let's see if it's going to work for him. The stacking stuff. Yeah, well, I was surprised at how easy it was, actually. So don't be jealous. Let's see. Oh, this is the link for the control room. It sends me to my own channel. Let's see. I don't think that's going to do it. Huh. Okay, we still got it. I will just prowl around and see what I can find out before I do the next one. Let's see. I got about the stacking stuff. Yeah, yeah. When you're out in the field, the spiders are just scurrying all over the place. And the way I've been able to do it is I have a colony in the lab and I just maintain them in the container. And oh, do I have them? No, I got them in the other room. Sorry, I got these nice little plastic, these lucite cages. They're about this big, really nice. And you keep the spider in there for a little while, and they build web there, and then they hang out in there. And these spiders are homebodies. So once they built their web, and you can just open up the lid, they'll just stay there, and you can just maneuver them around however you want to photograph them. Yeah, Dolomites is big. I haven't seen any around here, but I'd have to get down by the river to find them. Yeah. As for the stacking is I thought at first it was going to be tricky. You'd have to line everything and all that kind of complicated stuff. But I found this software. What's it called? It's called Focus Stacker for the Mac. That's an obvious name. And it's really easy, and it does automatic alignment, everything. You just drag a bunch of photos over there, and if they're at least close in orientation, it just neatly lines them up for you and stacks the most focused parts and produces an image for you. So it works out pretty well. Yeah, to find Dolomites, you gotta go near a river. We've got a nice big river over here, but there's no point in me doing it now because if I showed you the outside, it is frozen out there. It's covered with snow. It's Minnesota in winter, so everything is dead out there. Okay. Yeah. So Jasper Den Ouden is asking about focus stacking. I do some focus stacking. I've been lately doing some focus stacking when I can, when I've got a nice static image. But yes, I need to collect Caribbean spiders. That's a good idea. No, all of my proposals are about local spiders, so no, I can't justify it. Okay, well, I started talking about spiders, you guys. I said I wouldn't talk about spiders, and you are the ones who dragged me into talking about it, so it's your fault. So I'm just going to stop there and say, it's been fun, everybody. Let's do it again sometime. And I will, for Nestle, I will look into figuring out how to share these and have multiple people involved. I just don't know how to do it here. Okay, talk to y'all later. Bye. Seriously, really. End stream. Bye.